Hi everyone, my name is Kenneth Huynh. I'm a sophomore at CU Denver and a current lab member of Ernest Laboratory. I'm here to talk to you a new tool to detect engineered biotin-related nuclear proteins. So let's talk about some of the background. So there are several methods in detecting genomic locations of chromatin binding proteins. One of the most common methods you may have heard of is CHIP, chromatin immunoprecipitation. CHIP allows one to look at precise genomic locations as well as look at where particular proteins may localize at specific DNA sites. There are several disadvantages to this. If you have a low cell count, you'll produce low signals and low yield, resulting in inconclusive data. In addition to this, the size of protein fragments that you may be investigating or the DNA fragments that you may be investigating are limited to the resolution that the data may produce, resulting in difficulty in trying to determine the exact binding site of a protein. The counterpart to this is a new method called cleavage under targets and tagmentation method, cut and tag. It's another method to localize proteins in chromatin and allows us to identify DNA binding sites for proteins of interest. There's several advantages to this. A lower cell number is required because it uses enzymatic activity of TN5, which I'll describe in a little bit. There's also a cut and tag that can be assisted with a monomer called monomeric streptavidin, otherwise known as MSA, to provide high specificity and affinity. I'll talk about what TN5 and MSA is later in the background slides. So the overarching goal of this is to develop a more specific and high affinity method to determine where engineered biotinylated nuclear proteins bind in chromatin. Specifically, we make a biotin binding version of TN5 that can be used for cut and tag when a protein of interest is engineered to be biotinylated. This protein is called monomeric streptavidin TN5. So I'm going to talk about the agenda and what you can expect. So I'll talk more about the background. What exactly is MSA? What is TN5 and how it's being utilized? And the AviTag and BiRA relationship. And then I'll talk about the core of this project, which is developing the two expression plasmids, a mammalian vector and a bacterial vector. And then afterwards, I'll talk about development of, I'll show you the full range protein that we've been able to develop and the purification process that is currently ongoing. So what is monomeric streptavidin? So Park and Lim from University of Buffalo developed a stable and high affinity streptavidin monomer. Streptavidin is naturally found in the environment and it has a high affinity to biotin. It's normally found as a tetramer. Because it is normally found as a tetramer, it commonly aggregates. And if you've worked with streptavidin before, anywhere it finds a biotin, it'll basically attach to it. And with this monomeric version of it, a monomer, it's small, it's stable, it's synthetic, and retains its high affinity and specificity. Okay, and then where does the TN5 play into this? So TN5 is a transposase enzyme that inserts a short DNA sequence into the genome wherever it binds. This short DNA sequence can later be detected by sequencing genomic DNA. So the monomeric streptavidin, wherever MSA binds, wherever the biotinylated protein may exist, TN5 will basically insert an oligo in that particular region, which will later sequence with genomic DNA and see where that particular region is, allowing us to map where a particular biotinylated protein may exist in the genome. In addition to this, you may have seen it is commonly used in ATAC-seq, and part of our project is trying to purify and isolate our product, which is monomeric streptavidin TN5. And we see here on the right, there's a lab that isolated the TN5 version using a very similar method, which I'll talk about towards the end of this presentation. We do a very similar method in trying to isolate the product. So what is the AviTag and BiRA relationship? AviTag is a commercially developed 15 amino acid sequence that is a target of BiRA. So whenever you're developing some sort of a vector, you have to have AviTagged, which is an engineered epitope inserted inside, whether it's the mammalian vector or the bacterial vector. In addition to this, BiRA, as I've mentioned, is a target. AviTag is a target of BiRA. So BiRA is the enzyme that covalently attaches biotin molecules to the AviTag peptide. So as I've mentioned earlier, MSA has a high affinity to biotin. And because of its high affinity to biotin, you can see the relationship where TN5 kicks in. TN5 inserts the oligo, which can be later be detected with genomic DNA sequencing. So we see that AviTag is engineered inside this, whether it's the bacterial or the mammalian version, and then BiRA produces biotin around that region, biotinylating that particular site, allowing MSA to detect it, and then TN5 to basically mark it. So now that we've talked about the background, hopefully some of the pieces of the puzzle have been able to connect. I'll talk about the development of two expression plasmids, the mammalian vector and the bacterial vector. So we've cloned the MSA GFP version, which is the mammalian. Basically, we've been able to make a flag tagged vector with GFP into it and synthetically sequence a MSA version and attach it in frame with GFP to make a fluorescent fusion protein. And with this, we've been able to transfect it into cells. And we see that in the first row, it was able to be expressed in cells. And with rows 2 to 3, essentially, GFP was detected with anti-flag tagged antibody. And we see that it's able to detect the MSA GFP fluorescence. Okay, now I'm going to talk about the cloning aspect of the bacterial vectors. There's really more than 3 trials, but fundamentally it does come down to 3 overarching vectors that we try to throw it into. The first one is trying to clone MS18-5 into a PEX and HIS, which is just a histone tagged vector. We tried moving that to a PET30, which is also a histone tagged vector. The first 2 trials we've done were not successful. We weren't able to detect it on detect any protein on Western blotting, and so therefore we went to a new method. So trial 3 is where the money's at. Essentially, we cloned 3x flag tagged MSA by transferring the MSA GFP that you saw earlier, extracting the MSA version and moving it to a PATN5 vector from Hanikoff lab. So PATN5 is actually a vector that is used in cut and tag, and the PA in that aspect is what we're using to what we're taking to remove and replace it with the MSA. So you can see down here are the constructs that we've been able to develop. Here's the PX, here's the PET30, and then this is our recent one and also the successful one that we're trying to work with. And as you can see here that basically we just take out the PA portion and throw in our MSA. And something to note is also the CBD site, which I'll talk about later, which is the chitin binding domain. So I've been able to confirm the expression of the full-length MSA-TN5 protein through Western blot and sequencing. So you can see here that we're expecting around 99.6 kilodaltons. And I think this issue, as you can see here, we do see that there is flag tagged product detected here. And I have a positive control, which is just the PATN5. These are just uninduced proteins. And we do see something around the 90s range. And this may be a bit misleading as it says 93 here and we have 99.6. And I think I just didn't run the gel long enough. At first, we're considering that this may not be the right product, but we also sent it to Quintara Bio to sequence it, and it is confirmed to be the right product. So we took this and then we've moved it to try to isolate the product basically. Okay, so now is really the test for trying to purify the protein and isolate it through the chitin binding method. The first thing that before we even practice with the real monomeric streptavidin that we're able to make previously, I practiced with PATN5, which is the protein ATN5 version that we purchased. There's several steps into this, but basically you'll induce PATN5, you'll lyse the cells, extract the clear lysate, treat it with DTT, and elute it. This method, progressively, you should see that you start with the clear lysate, and then you wash it to get rid of any other gunk. You have it connected to resin and beads, and then you basically elute out the product. And with this particular part of the experiment, this trial, I've been able to basically uh, cleave off some of the product of PA, but not all of it. So we see that there's 2 bands here, and I went back to the drawing board and basically figured out what I need to do to make this a successful purification. So we're still currently working on that, and this is our most recent one, which we did recently in April. We adjusted to the Hanikoff protein purification protocols, which include a 48-hour cleavage as well as higher DTT, which the higher DTT, instead of a 50 millimolar, we're using 100 millimolar. And so this is a flag tagged Western blotting trial. And you can see here that on the darker exposure, which is where most of the data that I'll talk about here is, so there was basically 2 trials that I inserted into this Western blot. There's the old one, which had a wash and then the eluate. I wasn't able to basically cleave off any of the protein. So my old cleavage uh, experiment wasn't successful. I'm currently trying to do it again with a new method. Not necessarily a new method, but just higher volume and most higher concentration, allowing it to be better detected and cleaved. So we can see it on a Western blot. So we have the onput, which is the clear lysate, some of the flow through. So this should theoretically later be cleaved off, and you should expect a post cleavage of maybe a little bit less. Um, it's very similar to the P18-5, as you can see here earlier. Let me go back to the other slide. Initially, you're expecting 102.6 kilodalton for P18-5, and then when you cleave off the chitin binding domain, you come up with a little less protein. So basically, you end up with the 96 now. There, PA and MSA are off by about 100 base pairs, so you're expecting something very similar to that. And the future goals of this is to prepare a sufficient amount of pure MSA-TN5 protein. And then we also want to test the protein for biotin binding. We want to make sure that the MSA actually works and is able to bind biotin. And in addition, we want to test the proteins for its ability to insert DNA sequences. This is where the TN5 comes into play. And in addition, we want to test the protein for localizing biotinylated proteins on chromatin in cells. And I would also like to give some time to acknowledge everyone at my lab. I joined last summer, and all these individuals have been helpful and supportive in trying to help me understand the entire field of research, what it has to offer, the rigor, and they've been helpful to follow up with the project and help accelerate the project. So I'm very thankful to them. Thank you very much for uh, going through this project and watching this video. I really appreciate your time. And in addition, I hope this sparks some motivation trying to discover new methods of detecting genomic locations. Thank you.